Okay, class, good morning. So, we're having another experiment in genetics and entitled Extracting Your Own DNA. Okay, class, so in this experiment class, we are going to explore on the process of extracting our own DNA. Then, there are the several procedures on conducting this, but when we are following the steps in a correct manner, we are all able to have a successful experiment. So, it is a very easy to extract DNA at home just using everyday kitchen supplies. Then, you can extract your own DNA from your saliva, and you can use any fruit or vegetable and you can find at home. And now let us explore the world of DNA. Okay, class, so the materials that are needed. Okay, class, lagyan natin ng 1 tablespoon ng salt ang water, and then mix it para ma-dissolve ang salt. Second step, lagyan natin ng 3 tablespoon ng salt ang water. 3 tablespoon. Tapos, in the separate glass, lagyan na natin ng salt water. Ito na po yung i-gargle natin. Step plus, gargle the salt water for 1 minute. Next step, add 1 drop of washing up liquid to the salt water and stir gently. Try not to create any bubbles. So, dahan-dahan lang po tayo, class, para hindi siya maka-create ng bubbles. Okay, class. So, in a separate cup, mix the isopropyl alcohol. Dahan-dahan lang po tayo natin na i-mix. Kita nyo ba, class? Then, wait 2 to 5 minutes. So, okay, class, dyan na natin makikita ang result. After 2 to 5 minutes, ito na po yung resulta ng DNA. Okay, class, so there are questions are composed of 3 parts. So, first is observation question. So, number 1 is, are there any step you can leave out the procedures and still get result? Number 2 is, what other sources of DNA could you use? And number 3 is, does it help you to recognize your DNA and what way? So, B1 is analyze question. So, number 1 is, what happens if the alcohol is not cold? Number 2 is, what other sources of DNA could you use? So B2 analysis question number 1 is, explain how does it happen? Number 2 is, do you get more DNA from using your chicks and the fruit, the leaves, or the seeds? Why do you think this is so? So C application question. So number 1 is, what's happening during the process? So number 2, why does the alcohol needs to be cold? Number 3, cold CSI team use this technique collect DNA for a legal purposes. Number 4, what techniques are used for forensic science or medicines?